the previous class, we looked at the DNA structure and we saw how it can store information and replicate based on complementary base pairing. And today we are going to focus on DNA replication process and the repair process. Okay, so to continue where we left off, so the DNA structure dictates the replication process, and Watson and Crick kind of proposed that replication of DNA is semi-conservative. Okay, so what does that mean? So when the DNA replicates, so if we have this double helix DNA, it kind of opens up when it replicates. That's the only way it can be copied. And so this you call as the template DNA. That's the original DNA from which it is going to be copied. And so the new DNA is going to be made if the template DNA, as we saw before, is GATCTATG, and this strand will be CATA, coming right there, and GATC. So the new DNA that is going to be made will be complement of this CATAGACC, and in this case, this is going to be GATCTATG. So when you have these new DNA being made, this is 50% is a new DNA, and the template DNA is 50%. That is what is referred to as semi-conservative. Okay, so this is both of these are template DNA. Half the same, conserved, half new is a semi-conservative. Okay, but you need experiment to prove the DNA replication is semi-conservative, and this one is done by a different scientist called Michelson and Stahl. Michelson and Stahl. So these group of these two scientists, they worked out an experiment by using the isotopes of nitrogen, N15, N15, stable isotopes of nitrogen. So when the stable isotopes are being used, the main thing that you're looking for is the difference in weight rather than any radiation that's coming out of this. Okay. So to differentiate, which ones are going to be copied and which ones are going to be new DNA and all that, you need to differentiate. So they grew the bacteria in a medium that contained N15 medium. And then they transferred to an N14 containing medium. So you may wonder, what is the difference between one neutron between N15 and N14? But the DNA has millions of nitrogens or billions or trillions of nitrogen. So it makes a big difference. One neutron times a million, that would be million or billion, will make a big difference. So every like at 20 minutes, they were collecting samples out of this. And they did a thing called a density gradient centrifugation. So when you do density gradient centrifugation, the DNA will band according to its mass or density. So the results of this is the heavy DNA is at the bottom. And when they copied this, and then the next one, a combination of N15 and N14 was in the middle. And so this is a heavy band, and this is a medium size band. And this one had both a medium size band and a light band. Okay. So we need to understand how this is happening. Okay. So they looked at alternate hypotheses. Okay. So the alternate hypotheses are one is conservative mode. So in the conservative mode, both the DNA strands, like if you have a DNA strand like this is the template. And when it is copied, so this template will remain the same, and a new DNA will be formed that is going to be totally new. Okay. So this is called conservative mode, and the other mode is called as a dispersive mode. So dispersive mode, the template is going to be like this, but when it replicates, each strand has a combination of both old and both new. So in this case, you have a mixture of old and new. So they are testing, what is really happening? Is it semi-conservative or conservative? So this experiment proved that when they start with N15 and N15, so this is both N15 and N15, that is what makes this heavy. So it separates into the template, and when it separates, so it has a combination of N15 and N14. Okay. So you have N15, N15, that's a template DNA, and N14, N14 as the new DNA, and that is what creates this medium band. Okay, so that showed this is semi-conservative like this. And in that case, this can also be a medium density band. Okay, so if you have the dispersive mode, this can also be medium density. If you have conservative mode, you expect to find one heavy band, one light band. That did not happen. Okay, so that is a heavy band, this is a light band, that did not happen. But the medium density band happened, so you cannot distinguish between these two. Okay. So then continuing on this replication process, so this is going to split into two, this is going to split into two. So the template is going to be 2 N15. It is coming from this original N15, and this template is going to be N14, and this template is going to be N14. Okay. When you grow in N14, this will have a N14 DNA incorporated, N14 incorporated, N14, and N14. So when you have all these combination here, so this too contributes to the light band. This one contributes to the medium density band. And so this contributes to a medium density band. Okay. So the question to you is how this dispersive mode would look like in the third generation or third sample. What do you expect to see in this sample if it is a dispersive mode? Anyone? It's on the paper. Do you all understand the light, medium, heavy band idea? Correct. I think Daniel has answered that it is a medium density is what you expect, like written right here. But they did not see medium. They saw a light band and a medium band. That is how they disproved that this is not a dispersive mode. So the idea of this whole experiment is to prove it, the DNA replication is semi-conservative, and that's the whole experiment. So when I ask questions, I want you to understand the concept of experiment, why they did, what they did, how they proved their hypothesis, and they disproved the alternate hypothesis. Okay, so that's the main idea. And so going further, anyone have questions so far? Everybody good? So I'm going to get straight into the replication process itself. So long as you understand, what's happened here, follow through these colors of how the replication is happening. And the bacteria replicates every 20 minutes. So that is how they were able to prove this. Okay. And I will have some questions about it later on in the square cap. So you should be able to answer those questions. Okay. So the next thing is the replication starts at so that's origin of replication this is a particular region in the DNA in bacteria has a circular chromosome and this has the DNA like that. And the origin of replication is a single place, circular DNA, a single origin of replication, okay? So in the case of eukaryotes, they have multiple linear chromosome. And they have multiple origin of replication because it's a long and complex. So the origin of replication is also many of them. So that is long and so all these are origin of replication. This has a multiple origin of replication. Okay. So that's a difference between the bacteria and eukaryotes. So what happens at this origin of replication is the DNA kind of opens up. And that's where the replication has to happen. And so this is called as a replication bubble. And if you separate the bubble into two, so this is a replication fork. And you can have two replication forks. So this is how you view the DNA. If it is going from 5' to 3', this is going 5' to 3'. So you study how the DNA replication works in this replication fork. Okay. Any question up to this point? Everybody clear? Good so far? See, this is how life continues. Without this DNA replication, there's no new cell, there's no new baby, there's no new organism, there's no new life. So that's why it's such an important process. And if any mistake happens, it leads to mutations that can mess with the cell reproduction, lead to cancer, or lead to a new species being born and creating variation. So that is something important to understand. Okay. So any question up to this point? Everybody good? Okay, no further questions. Let's go to the actual process itself. So the DNA replication process, I'm going to go through the steps involved and the enzymes and what they do. Okay. So we started with origin of replication, and the DNA is relaxed by the enzyme called topoisomerase. Okay, the topoisomerase, it relaxes supercoiled DNA. Okay, to start replication. So the supercoiled DNA is all like an entangled web, and this is called as a chromatin. So this is what loosely dispersed DNA, and this is happening in the S phase of the interphase. So when it relaxes, it just opens up the segments of the DNA, and now it is available to do the replication. Okay. So the next enzyme that comes in is helicase. So the helicase enzyme unwinds the double helix. So when it unwinds a double helix, this is where, you have this DNA opens up like this, and that's where you create this replication bubble. Okay. So you wonder, the DNA has a hydrogen bond, the bases are nitrogenous bases, they need to hold the DNA together. So to prevent this DNA going back together, the SSB, which stands for single-stranded DNA binding protein, single-stranded DNA binding proteins, they stabilize the single-stranded DNA. So they kind of attach themselves to the single-stranded region, and they will avoid this from going back to a double helix form. So it has to be accessible to that, okay? And there is an enzyme called primase, and the primase, it makes RNA primer. And RNA primer is nothing but short fragments of single-stranded RNA, short fragments of single-stranded RNA. So when RNA primer is bound to that region, so the RNA primer, they bind to the template DNA, and they start to make the, I think this arrow goes like this. So these are the RNA primers. Okay, so you can see those short fragments, and I'm going to make this diagram bigger, so you understand. Okay. So the DNA that is the template, it's a kind of an open up like this, and so I'm going to continue like that. So that's your replication bubble, and it is open right there. Okay. So within this, what has happened is a single-stranded DNA is bound all around. Yeah. And the enzyme that opened up the DNA, we call us the helicase. So I'm going to label everything here, is going like this. So that is the helicase, and the blue is, these are the single-stranded DNA binding proteins. And the primers are attached to the DNA here, and they go in opposite orientation. So that is how the primers are going to bind. So let me label these here. So this is the RNA primer. Okay. So the synthesis of RNA doesn't need a primer, but synthesis of DNA needs a primer. It's the nature of the enzyme. That's how they replicate. Okay. So the next enzyme that comes in is called DNA polymerase 3. So DNA polymerase 3 has 3 functions. It makes DNA. It proofreads new DNA that was made, and it repairs any mistakes. It's an important secret of life. It's like a blueprint of life. If you make any mistake, it's a problem with the offspring. So it has all these functions. Okay. So this repair mechanism is what is called as the mismatch repair. This is done by this enzyme itself. Okay. So I'm going to zoom in on this same DNA. So now, but I'm going to keep it at the replication fork, and I'm not going to go into the bubble. So if okay, so so one is 5 prime going through this all the way to 3 prime, and this is the 3 prime to 5 prime. Okay, so the DNA that started with this RNA primer, and this is the other RNA primer. Now you have the new DNA being made, and so let me use a different color thing. We'll stick with this red, so that, but I'm going to do a wiggly line so that you understand the new DNA being made. The arrow is the RNA primer, and the wiggly line is the new DNA. So that's the new DNA. This is semi-conservative mode. It's a new DNA. Okay. Every time you need an RNA primer to start. So if the next stage, what happens here is same. I'm going to do this, the 5 prime to 3 prime strand. Now this opens up even more. That's a 5 prime to 3 prime. The opposite strand goes 5 prime to 3 prime in this direction, and the new DNA has to be made 5 prime to 3 prime, but it's a binding kind of complementarily to the other DNA. So this strand kind of continues, whereas whatever started at the bottom has to start with another RNA primer here, and that is what is happening here. Okay, another RNA primer. So the strand on the top, this is being continuously made, and you call this as the leading strand. That is continuously made. This is important to understand. Continuously made without any, as the DNA replication. So this is the direction of DNA replication. So this goes parallel to the DNA replication, whereas these are called as lagging strand, and that is discontinuously made. So it is not made continuously, and these two fragments are also called as Okazaki fragments based on the scientists who discovered them. So one strand, in this case, made continuously, another strand is made discontinuously, because that's where the nature of topology of DNA is made like that. Okay, and it continues. It is the same. The leading strand will maintain that way. So the next step is the DNA polymerase 1 comes in. This removes RNA primer because you want to make completely DNA, and replaces it with DNA. So this is where all these RNA primer that is shown here, that is replaced with the DNA. And the last but one enzyme is called DNA ligase that comes in, and that connects two fragments of DNA. It can be many the fragments of DNA. So this is where the ligase will come in, and I'm going to mark it up here. So this is where the DNA polymerase 1 will come and break this. and the ligase will come and connect these fragments. Wherever there is a gap, it is going to connect these fragments. Okay. And the last is gyrase. It's another form of topoisomerase. It introduces supercoiling to the DNA. So now they compact again, them relaxed DNA, they become more compact and go back to the chromatin. Okay. So any questions so far? This is a very important process. You need to know what enzymes do what. And many of the enzymes are named very logical manner. So once you understand the logic, easy to remember. Okay, let me go through that. Topoisomerase. Topoisomerase changes the topology or the shape of this enzyme, and it creates an isomer of the topology from very compacted supercoiled DNA into a relaxed DNA. So that changes the topology, topoisomerase. Helicase, it unwinds a double helix. So that's like a DNA is degrade DNA. This one opens up the double helix, and single-stranded DNA binding protein, it's self-explanatory. Primase, it makes the RNA primer. That's how you remember. DNA polymerase makes a DNA polymer. That's the enzyme that makes DNA polymer. 3 has 3 functions, and DNA polymerase 1 has 1 function to remove RNA primer to replace with DNA. Ligase, ligation means connection, and that connects to fragments of DNA by phosphodiester linkage. So this is the covalent bond that connects the 2 nucleotides together, phosphodiester linkage. And gyrase, like gyration or spinning, is where it creates a supercoil. Okay, everybody good so far? Any question? Almost done. We're wrapping up with the repair, and we are good. Okay. So the DNA repair, there are 3 types of repair. We already looked at 1 repair. So DNA repair mechanisms. Number 1 is mismatch repair. And that happens during S phase, during the replication phase, by the DNA polymerase 3. Okay. So there's a mistake, it fixes it. And the next type of repair is called excision repair. Excision repair is during the G1 and the G2 phases. After the DNA replication is done, this is where the DNA polymerase 2 comes in. And also endonuclease and ligase. All these enzymes come in, like if there's a DNA is damaged by UV, like happen in the summer, you get a skin and sunburn on the skin peels off because it's not able to repair itself. It's too much damage. And the last but not the least is called telomere repair. The telomere is at the tip of the chromosome, and it's difficult to fix it, because if you have, this is the tip of the chromosome, is a telomere, and you have an RNA primer that is attached to make the new DNA. So when the DNA polymerase 1 comes in, removes this RNA primer, there is no place to come back from to complete. So this part is also degraded. The telomere gets short and short and short. Okay. And this is without any telomerase enzyme, that would not be able to protect it. But with telomerase, if this is a telomere, this is a telomere, what it does is the telomerase enzyme comes, this is telomerase enzyme. So it will allow the DNA to be extended further region. So this is the 3' end, this is the 3' end. So the extended region is where the RNA primer binds, and it starts to, it makes the DNA, new DNA. And after this is done, the RNA primer is removed, the extended region is removed, so it protects the telomere. Okay? The telomerase enzyme protects the telomere region. That's also during S phase. We are done.